Hello, my name is Giullia Cazzanelli. I'm a PhD student in the Marie Curie Initial Training Netwok called GLYCOPHARM. Our network investigates a particular class of protein called galactins, which have the ability to bind to sugars. In particular, every recruited researcher focuses on different points of view of the same story, aiming to complement them, and finally, to unveil the significance of this protein in clinical processes and to develop galactin inhibitors and galactin mimetics.
I came from Italy and now I am a student in the PhD program on molecular and environmental biology in the University of Minho in Braga, a city in the north of Portugal. My task in the network is to use the yeast Saccharomyces cerevisiae as a model to study galactins, in particular galactin 3. Saccharomyces cerevisiae is also known as baker yeast or brewer's yeast, so now it's pretty clear that I was talking of the same yeast that is used to prepare bread and pizza, as well as, of course, beer and wine. What not so many people know, on the other hand, is that Saccharomyces cerevisiae is an excellent model organism, not only to study microorganisms, but also to investigate mammalian and specifically human protein. So here you can see the incubator in which my yeast grows at 30 degrees, both in solid medium and in liquid medium. These are the different medium used for the yeast growth and maintenance. And this, as you can see, is a burner Bunsen, necessary to maintain sterile condition while working with yeast. Saccharomyces cerevisiae DNA is easily manipulated. My purpose is to insert galactin 3 DNA inside the plasmid in the yeast and then make sure that the human protein is expressed in Saccharomyces cerevisiae in order to be able to analyze the yeast behavior and therefore to better understand the role of galactin 3. Another side of my project is to analyze what happens to the yeast when it's treated with the galactins from the outside. In this case, I'm going to observe at the microscope the morphology and the characteristic of the treated yeast, as well as some analysis at the flow cytometry, which is this equipment, very useful to understand if the yeast is still healthy after the treatment, if its membrane is compromised, and if the production of reactive oxygen species is growing, which means the yeast is not so well anymore.
In conclusion, I wanted to say how great is the opportunity to be part of a Marie Curie ITN. Indeed, beside the scientific part, which alone would be pretty good, since you have the opportunity to go to courses and congresses and to meet on a regular basis with the other member of your network, allowing you to learn stuff of which you barely know the existence, at least this was my case at first. There is the personal side, and you have the chance to live in a country that is not yours, to learn their culture and their traditions and their cuisine and sometimes their language, even if it's not always the case. Well, that's pretty good. So I hope I didn't bore you too much, and now it's time to say goodbye.